And this is on YouTube.
Okay.
On the Dixon YouTubes, okay. So what we just looked at was the anterior gray horn, where the cell bodies for the motor neurons are. Motor neurons are the neurons that go to the skeletal muscles, in this case. Okay. So this is going to a skeletal muscle. So what we are looking at is this right here. So, okay, so this is the cell body. This is the axon. You never have more than one axon per cell. The least number is 0. There's a few cells that have no axon, but most cells have 1, and you never have more than 1. What are these?
Other axons.
No, other
dendrites.
These are not dendrites. These are axon terminals and synaptic end bulbs from other neurons coming and ending on this one. Okay? These are the dendrites, because you can see they're continuous with the body, right? So this is an axodendritic synapse, axosomatic, axonic. This has more influence, everything else being equal, because it's near the axon hillock, where we start the action potential.
What did you call this one? Axo what?
Axonic.
Axonic?
There's an axon ending on an axon.
Oh, because these are on, yeah.
Okay. So messages usually come in through the dendrites, but they can come in through the body or the axons. But these are not real action potentials. These are called graded potentials. They can die out. It's like a coin dropping in water, and you get the little, waves. But those can die out. They lessen as they get further and further away from where they first came. So this causes a slight depolarization, but it doesn't fully cause a total depolarization and switch of the polarity. It's only here that if we exceed the threshold, if we have the voltage change enough, that we get a true action potential. So you only get real action potentials on the axons.
So this connection, this synapse, that's only where, I don't really understand that, what you're trying to say.
We only have action potentials that initiate here, at the axon.
So this is the sum. Just before that is the sum of all
Yeah, everything has to add up here to exceed the threshold. If it doesn't exceed the threshold, if we don't change the voltage sufficiently here, we don't get an action potential. Before then, it's called a graded potential. It's just a local depolarization. It's not a full depolarization.
So where these axons are coming in, so this is the only one that changes?
No, all of them do that. This has more effect.
Okay.
That's, again, a coin dropping in water, you get little waves.
Sum of all two.
So from here, the waves are fainter, right? Yeah.
So the waves, they die out a little further away you are from here.
So if you only have one action potential, it's not going to make a...
Now you're getting mixed up.
Yeah, I know, I am getting mixed up.
Yeah, don't get everybody else mixed up. Let me try again. Okay. These are called graded potentials before they reach here. These are not real action potentials. They're called graded. They can die out. It's only if you exceed the threshold here that you get a true action potential. A true action potential is all or none. It either happens or it doesn't happen.
So a true action potential can come from any spot?
No.
No, okay.
It's out at, well, these are graded potentials. If they add up to exceed the threshold.
If they add up?
Yes.
That's the
So one can't just make it there?
Probably
There has
not.
to be a whole bunch.
Probably not. Okay.
Okay.
It's confusing.
Okay. So it all depends on whether you exceed the threshold. If you exceed the threshold here, then you get an action potential. Travels down. Okay. Now we have things here. What's this blue with the red? That looks like our old friend Ruffy R, right?
Yes.
But it has a special name here. It's called Nissl bodies, or chromatophilic substance.
Nissl is an N or
N-I-S-S-L. Okay. There's also lysosomes here. I'm not sure if those are yellow or not. But the lysosomal wastes are called lipofusin. It's sort of a yellowish brown pigment that accumulates. I call it lysosomal poop. Okay. There's also some Golgi here, you can see. Some smoothie R. Here's a nucleus, here's a nucleolus. So basically it looks like a regular cell in here, except something's missing. What?
centrioles.
No centrioles, because these don't divide. You can get some neurons formed in some parts of the body from neural stem cells, like in the hippocampus. The hippocampus is involved in memory. And you do get some cells there under certain circumstances, like when you really stress to learn something, that's when the cells stick around. If you don't, they stress them, they come and they go. So This class is working to make the new neurons.
Yeah.
Okay. So the action potential travels down here. Now this is the axon part, okay? And then at the end, it'll look something like this. Now this happens to be a myelinated neuron. What kind of cell is this? This is a Schwann cell, or neurolemmacyte. And notice how it's very tightly wound around, like a jelly roll. The inner part is called the myelin sheath. It's basically phospholipids, sort of like a plasma membrane. It's been tightly wrapped around. And some of the stuff gets pushed to the outside. And that's the nucleus and some of the cytoplasm, and that's called the neurolemma. Neurolemmas are important because they help guide an axon if it has to repair. Now, is this unipolar, bipolar, or multipolar? Unipolar?
Multi.
Oh, I was thinking of out. You're saying in. That's right.
Okay, what is the definition of unipolar, bipolar, and multipolar?
Incoming.
This is one process.
How many processes do you
Yeah,
have?
one process.
Yeah.
One process coming in, right?
Well, how many processes are sticking out of the cell body? How many processes do you see sticking out of that cell body?
Lots.
So it's
Yeah, well, this is multipolar,
Okay, so never mind in or out, just in general.
in general.
Ah, okay.
Processes
Okay.
sticking out,
Okay.
some in, some out, okay?
Okay.
But they're sticking out, they're all sticking out.
Yeah.
That's it. Okay.
this is-
If you only have one, then you're unipolar. Unipolar are the kind that come in carrying sensory information. These are unipolar. Bipolar are you don't find them in many places, the eye, the ear, and the nose. Association neurons are usually multipolar. Motor neurons are multipolar. Okay, so this is a multipolar neuron. We know this is not an association neuron because all association neurons are in the central nervous system. And in the central nervous system, we myelinate with the oligodendrocytes, not Schwann cells. Since this is using Schwann cells to myelinate, this has to be in the peripheral nervous system. So this starts in the central nervous system, in the anterior gray horn, and then it comes out and goes to skeletal muscle. What's this covering called?
No.
No,
Is this the neuron?
Yes, it's endonurium.
That's right. This is the lowest one. Yeah.
Endonurium. So these are axon terminals, synaptic bulbs. Are these touching? They are here, but are they
There's a
really
gap.
touching?
there's a
No,
cleft.
there is a little synaptic cleft, okay? And so, and then are these all excitatory? Are they all causing? Nope, not necessarily. They could be excitatory, or they could be inhibitory, or it could be a combination.
Right. That's right.
So that's why it all depends on how it sums up here, whether we get a real action potential.
Still confused.
Okay?
How do you know if it sums up there?
If it sums up there and sees the voltage change, then you get an action potential. If it doesn't, you don't get an action
Do
potential.
they have a specific voltage potential? Do they know the gate, the voltage potential of the gate? What is that?
Usually it's about minus 55 millivolts.
Okay. Minus 55. Oh, because it's negative, duh.
Yeah. Well,
okay.
across the membrane,
Yeah.
there's a difference across the membrane. It's a little bit positive outside, a little negative inside. Often it might be a difference of minus 70 millivolts, sometimes more. But if we get up to minus 55, then that's
So this could be minus 25, this could be...
No, they're all the same.
8.
Oh, on the membrane, it's all the same.
No, I meant, okay, so I meant the charge coming in.
Oh,
So the charge coming in could be vary. It could be
Well,
all
like I
kinds of
said,
weaker...
you could be opening up sodium gates, sodium
Yeah.
rushes in.
Or
that
chlorine gates to inhibit.
Or, yeah, chloride gates opening up would inhibit. Okay.
Okay.
And I can give you a handout on this if you want later on.
Yes, please.
And we can watch a video on this too if you want. And the best thing though is the interactive physiology down in the tutoring room. Interactive physiology is very good on this. Not interactive anatomy, it's interactive physiology. And they have something on the action potential, and they have a whole number of the one on synapses, but the action potential is probably what you want to look at.
Okay.
And has anybody looked at that?